international scientific research project that aim to determine the complete sequence of nucleotide base pairs that makes up the human DNA. So one thing I would like to make clear over here, so it is nothing like, it is not that you had selected one person or uska pura DNA it was sequenced. No, it is not that way. It is like in human beings, all the genes that are present. So 99% of the genes are same, right? So they are going to be sequenced. Along with that, whatever the variations were there, so those variations would also be sequenced. Okay, so it's one of the largest collaborative biological project. That collaborative in the sense, like many different countries had come together, many different research labs all from all over the world had come together to collaborate in this particular project. Okay, so the idea was first picked up in 1948 by the US government. Okay, they started to plan it, and then formally it was launched in 1990, and the final whatever the results that were completed and that were out, that were made public in 2003. Okay, so originally it aimed at mapping the nucleotides contained in a human haploid reference genome. Okay, so when I say human haploid reference genomes, means one set of genome that is present in the humans containing all the genes that could be possibly present in any man. So the genome of any given individual is unique. Okay, that is what we were talking about. So mapping the human genome involves sequencing the genomes of small number of individuals and then assembling them together to get a complete sequence for each chromosome. So after this project, we also came to know that which genes are positioned at which chromosome at what niche. Okay, so every chromosome will have different parts or different positions, right? So at which position a particular gene is present, that can be found out. So the finished genome, human genome does a mosaic. Why do you call it as mosaic? Because it is not representing one individual. So when the DNAs were randomly selected or cells were randomly selected, the subjects were randomly selected. So it was taken care that it was all the samples were representative of different races and different creeds from all over the world, right? You understand that we are Asians. Asians maybe we have different ethnicity, like we are South Asians are little different. Like you go towards little east, like your China and all, they have a different kind of an ethnicity. We will have a different kind of ethnicity. The Western countries, which are called as your Caucasians, they will have a different kind of ethnicity. And the variable genes which are present, that is going to be a little different with respect to this race and our ethnic, whatever our ethnic background is there. Okay. So that is the reason why this human genome which was mapped, it would be considered as a mosaic. So what are the goals of this human genome project? It was to identify and map all the 20 to 25,000 genes approximately which is present in the human DNA. So finding the functional point of view as well as the physical. So I told you that mapping will tell us that at what position these genes are present, and also here we were not just focusing upon the physical map, we were also wanted to know what were the functions of each of these genes that were determined. Okay, so to determine the sequences of 3 billion chemical base pairs that make up the human DNA, to store this information in the databases. So whatever, once you have found out the sequences, the information would be stored in the databases. To discover more efficient technologies for data analysis, like just getting the sequence is not going to help us. We have to know specific regions. Okay, this particular sequence is for this gene, and this gene is responsible for this function. So these kind of your data analysis was important. So allow the private technology that arise from this project. So this human genome project could be taken as the basis of many upcoming projects. Okay, so that was that was one of the aims that was taken care of, that it will form the basis, and they did not want to keep this. So there was again a lot of some people thought that we should patent this, or so the information should not be out and all those things. But there were some people who thought that it is important that this information about the human genome, so it should be out, it should be public. Then all the scientists all from all over the world would be able to utilize this information to develop more better technologies. Okay, so that was one of the goals. Okay, so also to sequence the genome of other organisms. Now this is also very important. So here it was not only focusing on human. So now today if we see the research going on, so mostly the research is going on for the development of the, for the betterment of human beings, but in that we have so many other animals also included. For example, your model organisms like your mouse, your Drosophila, and a lot we are coming to know from their genome, right? A lot of like E. coli, C. elegans. Yes, so there are so many types of your model organisms. So studying them is equally important. So until and unless, and studying them is easier as compared to humans, right? So they tried to find out the sequences of these model organisms as well, so that we can use those information for understanding the role of different genes in humans. Okay, so knowing these genomes of these organisms was also one of the goals of this Human Genome Project. So to address ethical, legal, and the social issues, because it's a DNA sequence, there could be so many different types of issues that will arising. So taking care of all those things, that was also one of the goals. So who were the sorry. So as I told you, that it was one of the largest collaborations. So there were many different agencies. So in 1990, it started with the two major funding agencies, that is the US Department of Energy and National Institute of Health. So they developed an MOU, that is Memorandum of Understanding, in order to coordinate the plans, set up the clock, that means do a project flow, कि ये year में इतना हो जाएगा, उतना year में. So like and please understand that in 1990s, sequencing and all was not very easy as it is today, because of the next generation sequencing and all that we have learned. Today the sequencing methods are very developed, and it becomes very easy to sequence an organism. Wouldn't take a lot of time, but way back in 1990s it was a difficult task. Definitely it was a very, very difficult task, and a lot of time consuming process as well. Okay, so many years would take for the sequencing of a single organism's genome. Okay, so that whatever the timeline of this project, that was decided. Most of the government sponsored sequencing was performed in 20 universities and research centers throughout the world, like US, UK, Japan, France, Germany, Canada, China. Okay, a parallel project was conducted outside the government sponsorship by the Celera Corporation, or the Celera Genomics, which was formally launched in 1998. Okay, so this was because they were not sure whether the information is going to be shared with the the private companies or not. So for development of that, so $3 billion project was formally launched in 1990. But as the years went on, a lot of more funds had to be collected. So the Human Genome Project was a 13-year-long, that is what I was just explaining to you, publicly funded project initiated in 1990 with the objective of determining the DNA sequences of the entire euchromatic human genome within 15 years. Okay, so let us now see that how this genome mapping was done. Okay, how this physical maps, genetic maps were determined, the technical aspects of this same. So genetic maps are invaluable for genome mapping. So markers are inherited. Yeah, sorry. So genetic markers are invaluable for genome mapping. So markers are any inherited physical or molecular characteristic that are different among individuals of a population. Okay, so that means they are polymorphic. All of us are human beings. So 99% of our genes are going to be same, but there are some genes, there are some markers which are going to be unique. Okay, that makes us individual of a particular population. So our population is polymorphic in nature. Different types of your genomic things are there. So genetic map shows the relative location of the specific markers, where they are present exactly. So even if you have an SNP, so genetic markers, you have RFLP, you have SNP, you have SLLP. Okay, different types of markers are there. You have this SSRs, that is short tandem STRs, tandem repeats. Okay, so which are also called as your microsatellites, right, which are present at the ends of the chromosomes, right? So such kind of your genetic markers are there. So finding out those genetic markers, so even if it is an SNP, which is just a single nucleotide polymorphism, still there is a position of the single nucleotide polymorphism. A specific map can be made to see where is the position of that SNP in a chromosome. So an example of a marker includes restriction fragment length polymorphisms, right? So what happens in restriction length polymorphism? From the name itself, you understand that the positioning of these restriction signals. Okay, so the enzymes recognize a particular signals, right? A sequence. So restriction sites, the position of this restriction sites is going to differ in different individuals. So that is the reason why if I take a sequence from individual 1, 2, 3, 4, say 4 individuals are there. If I take a specific part of a sequence, okay, say samjho, I am taking a region from P arm of chromosome number 1. Okay, charo ka. So if I am restricting it with the same enzyme, when I run the gel, I am going to get different band patterns, because each of these 4 individuals are going to have these restriction sites at different locations. Okay, they are going to have this at different locations. Okay, so are you understanding the what is RFLP? A quick yes or no, or you want me to explain it in much more depth? Okay, so what happens is, I'll let me show you using a pen, right? So say I have 4 different individual ka DNA. Okay, so it is from the same sites. Okay, in each of these people. So now what happens is, I'm now marking the restriction sites. So restriction site, for example, Hindi 3. So in this particular DNA sequence, it could be at this position, it could be at this position. In this individual, it could be at this position only. In this individual, it could be at this position, it could be at this position, it could be at this position. In this individual, it could be somewhere here and somewhere here. So what happens? When I get these sequences, in this, I'm getting these are my fragments. These would be the size of the fragments for this individual. This would be the size of the fragments for this individual, right? I'm going to get 4 over here. Right here, I'm going to get, this is a bigger fragment, this is a small, and again a small. So see the fragment sizes, see the fragments numbers, sizes. They differ from each other, right? They are differing from each other. So if I have a gel, so in this gel, if I run 1, 2, 3, 4, so I am going to get bands at different positioning. So depending upon how many bands I have got, and depending upon what is the length of my DNA fragments that I have got, right? So you will be getting as a alag-alag banding patterns. Okay, so this banding pattern suggests the RFLP profiling of profile of a particular individual. Okay, so that is how it is. It can be used as a genetic marker. Okay. Yes, everyone understood? A quick yes or no? Okay. So now let me go forward. So that is about technically which were the how we went on. We did not go. We specifically talked about the genetic markers. Then to sequence DNA, first it was amplified so that the quantity was increased. Two types of DNA amplifications are cloning and PCR. I'm not going to depth in that. And all these things you have already known. Okay. So DNA has been amplified. Sequencing can be done. So sequencing was done by shotgun sequencing method and Sanger sequencing method. Okay. So shotgun sequencing is nothing but you are going to, so this is your genomic DNA. So by shotgun, that means it's nothing but where you are fragmenting it. Okay, shotgun is nothing but fragmenting. So the entire genome is going to be fragmented. So if you see that they are randomly fragmented and every gene is going to have one or the other overlapping regions. Yes or no? You can see over here that this and this region is overlapping. This and this region is overlapping. Here also overlapping. So every fragment is going to have some or the other overlapping regions with other fragment. Okay. So then they would be sequenced like this. And once you get your sequences, then they are going to be assembled into the entire genome. So how do you assemble it? Definitely by knowing the overlapping regions. Okay, so overlapping regions for again, vapas using your softwares, they would be aligned. Okay, so they would be aligned together using different softwares, and that is how the entire sequence would be deducted from this short whatever your sequences are there. And Sanger's method, I don't have to explain you again. You already know about the Sanger's method. Okay, so you know that you have 4 different reactions. Okay, and each specifically reacting to a specific type of your DNTPs. And then from the gel that you are going to get, you can read what the sequence would be. Okay, so this was used for a low standard. So shotgun method is preferred as for a large human DNA, it is easier for you to carry out this process. Okay, so this is just the overview of Sanger's sequencing. So as I have told you, and at that particular time, they had already started with the automatic Sanger sequencing method. So here you can see the automatic method where fluorescent dyes are being utilized for the same. Okay, so what was the outcome of this human genome project? So approximately 22,300 protein coding genes in the human beings. The same range in the other mammals, like around 23,000 genes approximately were found out. Then Drosophila around 17,000, then C. elegans or less than 22,000. So here one thing is very astonishing, don't you think so? That even if we are human beings are large, we are big animal as compared to mouse or Drosophila or C. elegans, they are very tiny organisms, but still the number of genes is very much comparable to them, right? Okay, so in case of mouse, and here you can see that the mouse is having much more, or the number of genes is more as compared to us, right? So why that is the case? Because of your junk DNAs, okay, or repeats, you can say. So it is not true that that as the organism becomes big, the number of genes increases. No, it is not, because some of the times you can have fused genes also. Okay, so we share a lot of homologous genes. Yes, orthologous, you know, or what is orthologous? That similar genes or similar proteins present in different animals. Okay, so many of our protein coding genes produce more than one protein product. Yes, that is what I was talking about. So on average, each of our 4 produces 2 to 3 different proteins. So that is the reason why even if our genes are less, the complexity of the proteome is much more than your Drosophila or your C. elegans. That means see, for example, in Drosophila, say one gene is giving rise to 2 proteins, but in human beings, the same gene may give rise to 4 proteins. So gene number kam rahega human beings mein, to bhi the number of proteins that have been produced in the humans is much more as compared to your these smaller organisms. So that will account for the complexity in the larger organisms like us. Okay, so you have to know that the genome size may be less in humans, but the proteome size is definitely more than these smaller organisms. So a larger portion of our genome codes for transcription factors, which controls these elements like, you know, enhancers, suppressors, and all those things, and also your transcription binding sites. So the combinatorial use of these elements provides such greater flexibility of gene expression as compared to your lower organisms like your Drosophila and your C. elegans. Okay. So we came to know about the gene density also, 23 genes per million base pairs on chromosome 19, 5 genes per million base pairs on chromosome 13. So human and presumably most vertebrates have genes not found in invertebrate animals like Drosophila and C. elegans. And few of those are like antibodies, your T cell receptors, your transplantations, antigens like your MHC, HLA complexes, right? So these genes are not found in the invertebrates. So sorry. So it was found out that the immune system, that is the reason, is much more developed in your vertebrates as compared to your invertebrates. Okay, so all these genes are absent in those invertebrates, but they are present in the vertebrates. Okay, so human genome comprises of 2% of exons. Okay, so amongst the entire bulk of your DNA which you have, only 2% of it is actually coding for the proteins. Rest is your non-coding regions. Rest will account for your introns. Okay, they are repeats or some other sequences. Okay, jump DNA you can say. So let us now move on to the actual part of this lecture, that is your applications. Okay, so I hope everyone is clear with the basic of Human Genome Project. Yes, everyone, a quick yes or no. Yes. Okay, great. So let me move forward. So basic application was definitely with respect to medicine. So the sequencing of human genome holds benefit for many fields, from your molecular medicine to human evolution. So understanding human evolution, which parts were more evolved, or where the human evolution, which like if you study from different races, from different ethnicity, it will give us hints like which part had started to evolve, which part of the world had started to evolve more and which started to evolve lesser, all those things. So helps in identifying which are the disease causing genes, identification of mutations linked to a specific form of cancers or any other. See, what happens is SNPs, that is a short nucleotide polymorphism, even if it is a change in a single nucleotide, but these SNPs have been linked to so many different types of your genetically caused diseases, like your cancer, like your say diabetes, or your any kind of your hypertension or coronary arterial diseases. Okay, so all these have been linked together, even if there is a single change in the nucleotides. Okay, so the sequence of the DNA is stored in the databases available to anyone on the internet, and then US National Center for Biological Information and its sister organizations like, you know, NCBI, you have your EMBL and DDBJ. So they all are storing these databases. Okay, you have this genomic informations. Okay, so in NCBI we have it as a gene bank along with the sequences of known and hypothetical genes and proteins. Okay, why do you call it hypothetical genes or proteins? Because where we may not have been able to find out the sequence as actual sequences, but maybe from a protein we have to try to deduce the gene sequence, or from the genes we try to deduce the protein sequences. So these Human Genome Project has allowed us for advances in agriculture through genetic modifications, so that today we are able to produce GMOs where disease-resistant crops or your any other environmental effects can be reduced. And definitely it has helped us in one of the major technologies in forensic sciences, that is your DNA fingerprinting. Okay, so it was a huge boost for that. So now let us just focus on the implications in medical science. So at this particular part, if you get a question, you will be getting what is the implications of human genome mapping in medical science. So from this, whatever the slides are there, so that is 3 to 4 slides, that is going to be asked, or that is going to be your answer for the same. Okay. So having the human genome sequence and knowing the DNA spelling variations among people will help reveal which genes contribute to the risk for common diseases. As I told you, that having a particular SNP may predispose us. Okay. So for example, let me give you an example of a gene called as your PPAR gene. Okay, so this particular gene, PPAR gene, has many different SNPs. So there are some SNPs which will predispose. So for example, I have 3-4 of my siblings are there. Okay, so I got one of the SNP which is predisposing me towards this diabetes. Okay, so my other 3 siblings, if they don't have those SNP, only I am having that SNP. Okay, I have inherited from maybe my father or my mother, but my other siblings have not inherited. So now what is going to happen is, even if 4 of us have the same lifestyle, say we are having a sedentary lifestyle, or 4 of us, but still because I am having that SNP, I am more at a risk of developing that particular disease. Okay, so if we could already find out this, we could already associate any kind of your genes or any kind of sequence for such kind of your common diseases, then it may help us to anticipate it prior. Matlab, before that diabetes onset takes place on that particular individual, the person will know that I am more prone to have the disease, so that person will take the proper precautions. Okay, so this is going to be a challenging task. So for example, researchers expect that 5 to 10 and perhaps more genes are involved. Yes, in diabetes there are a lot many genes which are involved, so finding out SNPs and combinations of these SNPs, how they interact, all these have to be studied. Okay, and how much is the risk factor, that has to be studied. And such kind of studies can be done in a huge population, or has to be done in a huge population before we link any gene to a particular disease. Okay, so again we have something called as your epigenesis or epigenetic factors. Okay, you must have learned about this. So what is epigenetic? Epigenetic is another, but even if you have a particular gene, okay, see, finally the function of the gene is being brought about by your proteins, right? The product of your genes is your proteins, and it is the proteins which bring about whatever the changes that are there in our body. Now, so that means the expression of the gene is very, very important. So even if I have a defaulter gene, say for example, but the environmental factors are such that there is some epigenetic faction and the expression doesn't take place. Okay, there is no expression. So what will happen? Again, because of the environmental factor, the gene interactions change, the expression patterns change, and again that is going to have a effect on that particular person. Okay, so these, and when I say about environmental factor, it is about your eating habits, it is about the like, that's what I'm saying, that even if I have that SNP, it may happen that I'm not developing the disease. The reason is the same, because my food habits may be very good, my I am regularly exercising, I don't have a sedentary lifestyle, so that has an, so such kind of environment will has an effect on the expression of that particular gene. And that is how it will, so the interaction of your gene with the environment is also a very important factor. So all these things have to be studied. So even so, because of all these problems, still researchers are optimistic that one day we would be able to diagnose different forms of diseases like your cancer, diabetes, heart, by developing a kind of a catalog in the sense a kind of a profile कि अगर ये genetic variations है, then you will definitely, or you are more prone to get this diseases. So ये बात पहले से patient को पता होनी चाहिए या patient को बता सके. That kind of a day will definitely evolve with these kind of advances that we are making in the medical science. Okay, so researchers will increasingly bring to light the molecular process that normally maintain the human body in good working order. So diseases, so studies will also reveal how the same process are disturbed in illnesses. Like what is the molecular basis of a particular disease? What exactly change is brought about in your cells or in your tissues that leads to these diseases? Understanding this has been helped by your Human Genome Project. Okay, so the discovery of such genes, genes that affect the risk of an illness, will reveal clue of how this illness arises. And once we know that how this illness has arisen, then it would be definitely easy for us to develop a response to that, to develop a drug to that, and also to study whether that drug has any effect. What is the variation? So what is the drug response? Whether the drug is effective over that illness or not. Okay, so ये सब चीजें genomics से हम study कर सकते हैं. So genomic is likely to help allow the prediction of individuals' responsiveness. So this is again something we are looking forward to, that is called as your personalized medicines. Okay, so today what happens is, like say I have individual 1 and individual 2. Both of them are having similar symptoms. They go to the doctor and doctor will give them similar medicines. Okay, but now both the individuals are taking the same medicine. One individual will get the effect very quickly, and maybe in the next week in the follow-up he comes and says, okay doctor, I'm absolutely fine, I'm doing good. But the other person will say no, the medicines were not effective, and maybe the person comes back after just 3 days and say it's not working out for me. So why does it happen, right? So because individual to individual, the metabolism is different. Okay, so why it is different? Because our genomics is different, right? The genome that these two individuals had are different. So their proteomic is different, metabolism is different. So how the drug is responding, that is going to be different. Okay, so if we as we are moving forward towards, so taking this Human Genome Project as the basic, we are looking forward to such kind of your possibilities, like where by studying the genomics of a particular person, we would be able to prescribe drugs according to their genome. Okay, so that is what is called as your pharmacogenomics. Okay, so that is going to, you would be having some part of it in the next semester. Okay, so here they would be in like trying to make a prescription of drugs which is more individualized or personalized for a particular person. Okay, so that is the future. That is the future of our medicines today. Then researcher will need to develop and apply methods that analyze many drugs at a time for their potential effect on disease-related genes and gene product. Pharmaceutical industry has been gearing up for this opportunity. So you can have, depending upon genomics, we will have many different drugs. Okay, you cannot go for a single set of drugs. There could be many different drugs that would be required. So definitely it is going to boost up the pharmaceutical industry. Okay, then efficient ways of analyzing the effect of this drug should identify those that block or stimulate particular genetic pathways. Okay, how if we can have a drug, so there is some genes which are being blocked, some expression which are being blocked because of some kind of your thing. So that can be reversed by some drugs. All these studies have to be decided. Okay, this can be used to treat cancers like leukemia. Yes, definitely this can be used to cancers and all. Yes, they are focusing on such kind of your genetic pathways only. Okay. So for example, let us see the development of drug STI 571. So these are just kind of generic, they are not the actual name of the drugs, but it is some identification number for the pharmaceutical industries. Okay. So which are designed to block the activity of your BCR-ABL gene. So BCR-ABL gene is a kind of a fusion product, okay, between your 9 and 22. Yes. So you have learned about this, right? You have Philadelphia chromosome in this. So this is one of the major reasons of your CML, that is chronic myelogenous leukemia. So if we are able to block the activity of this fusion gene, then the fusion protein will not be produced, or the so what happens here is the genes are overexpressed. Okay, so that overexpression can be blocked. Okay, and this, then leukemia will not be taking place. So it will block the activity of the BCR-ABL product to chemically modify an unknown molecule. So the drug has shown dramatic results in early clinical trials on the patients with advanced CML. So that is how we are trying to reduce the effect. We are trying to convert or bring back whatever these genetic changes are there, reverse these genetic changes which are there. Okay. So these genomics will definitely help in prediction of how an individual reacts to a particular drug. So this is just the same example that I've talked about. So I'm skipping the same. So the field of gene therapy has again sustained a series of disappointments over the past few years and has gone back to the basic scientific questions. So major questions is safety, right? How safe it is. Yes, transferring genes in the human body. Later on, because we don't know exactly how a gene may react once it goes inside the human body. Okay, so safer and more effective ways of transferring genes have to be developed, and in the next part of this particular unit we are going to learn that genome editing, what are the different ways and how promising it is, and how exactly we are today definitely using those methods. Okay, so we will discuss that in more in the subsequent modules. So predictive genetic tests are likely to be available for more common conditions, allowing individuals who wish to know this information to learn what their individual susceptibilities are, to take steps to reduce those risks for which interventions are available. I have already explained you all these things, so I'm just reading this. So the interventions could take the form of medical surveillance, lifestyle modification, change in diet, or drug therapy. So for example, if a person already knows that he is more susceptible to colon cancer, so frequently he will go for colonoscopy for seeing the screening, because in cancer, one of the major problem is that it is detected at a very later stages, and by the time it has already started to spread in your body, right? So that's one of the major problems. So all these things could be taken care of by this predictive genetic test. Okay, so that, and if I know that I have a family history, so again it becomes much more simpler. Okay, much more easier to screen. Okay, but definitely there are some problems with increasing genetic information available about common illnesses. Genetic risk assessment will become more generally available. So primary care providers, that means our normal doctors, physicians, will need to practice genomic medicines also. So today that is the major problem in India, at least. Okay, if you see in India, the whatever practitioners are there, they don't have an idea what is going on in the research field. But that is not the case if you go in abroad, like in US or UK. There the doctors who are practitioners, they are also involved in research. But that is not the case in India. That's the problem, that they really don't know much of genomics, and it is like paramedical people like you and me who are more having knowledge about these things. So it requires a real collaboration between these primary healthcare workers and the paramedics like us, or people who are having information or knowledge about genomics, to work hand in hand. And a lot of statistical, as I told you, that all the studies need to go in a larger population. So a lot of statistical studies will involve. See, that is the essence. It is not that when you want to do something great, it is not that as only a biologist I can do something. Yes, it has to be an amalgamation of work from various different people. It has to be collaborative. So that is the reason why a biologist also has to. Why do you learn biostatistics? Okay, you are learning that. I have already told you that it's very, very important for a biologist to know statistics. Then only he or she would be able to find out what is the data talking about, whether it has any significance or not, right? So this will require substantial advances in understanding of genetics by the healthcare providers as well, as I told you. Then again, the problem comes about the privacy of data, because data is a huge thing. A lot of money making today, you already know that. And when you have it as a genetic data, a lot of privacy has to be maintained, right? So a lot of legislation or has to be passed on that bans the use of genetic information that predicts future risk and decision about health insurance and your employment also. Like if it is available freely, so what will happen? Employment जब से हम इसके लिए जाते हैं, इंटरव्यू के लिए जाते हैं, if it is available online, so that person will search, the interviewer will just search the genetic of this person and try to find out whether he is more prone to any kind of such kind of your lifestyle diseases like your diabetes, cancer. Because what happens is, it is a liability for that company, right? It is going to be a liability for that company, and the company will say, he is prone to these diseases, so I think we should not take him for this job. So it will be, so there are these kind of things also which has to be taken care of if such kind of your prediction of depending upon our genomics is publicly available. So such kind of things will definitely arise, okay? And in health insurances also, you know that depending upon your history of medical history and all, the insurance companies also decide that what should be your EMI and how much they can support. Yes, all these things will differ. So it would be very difficult for such a kind of a person who is whose genomics already says that you are prone to these diseases. Then definitely these insurance companies will not be ready to insure them. Yes, and whereas they need the most, right? So all these problems are going to be there. So that will lead to discrimination, a lot of discrimination. So by 2020, the impact should be more. That is what was foreseen, okay? But still today, pharmacogenomics is, we are trying to understand pharmacogenomics still, right? We are trying to understand the predicting of the drug responsiveness towards or depending upon our genomics. Okay, so we are trying to still find out whether designer drugs is a reality or can be a reality or not, okay? So a lot of different diseases are there, which in the future, we can definitely think of. So diagnosis and treatment of cancer will likely to be transformed. Yes, definitely today, it is not that it is not transformed. In cancer treatment also, a lot of molecular fingerprinting, a lot of finding out of, where exactly the, or what is the exact mutation that has brought about the development of a particular tumor. These studies have already started. Okay, these studies have already started, but still we can't say that it is in full swing or it is the only thing that is dependent upon. We still are dependent upon your chemotherapy and other methods also. Okay, but in near future we can definitely think about all those ways. So will there be just a second, let's stop.